So what is transcription and why is it necessary? Transcription is the first step in the process of using genetic code in DNA to synthesize or build all the different proteins in your body. One problem with synthesizing these proteins is that the instructions for making them are in the DNA, which is located in the nucleus. The place where proteins are made is outside the nucleus, either in the ribosomes floating in the cytoplasm or in the ribosomes embedded in the rough ER. How does the genetic code for synthesizing proteins get from the DNA to the ribosome? DNA uses a messenger called messenger RNA or mRNA to carry the genetic code from the nucleus to the ribosome. The process of building this messenger's RNA is called transcription. How does transcription happen? Transcription begins when an enzyme called RNA polymerase attaches to a segment of DNA called a gene. Gene is a section of DNA that provides the instructions to make DNA. A gene contains the code to build a specific protein, a macromolecule made up of a sequence of amino acids in a specific order, such as catalase. Within a gene, the specific order of nitrogenous bases dictates the order of amino acids that will make up a protein. RNA polymerase causes a particular area of a DNA helix to unwind and separate into the two strands. The non-coding strand is the side of DNA that is read or transcribed by the messenger RNA. The non-coding strand is the 3' to 5', meaning the 3' carbon faces up in each sugar and 5' carbon is below. The other strand of DNA, often called the coding strand, isn't transcribed by the messenger RNA. The coding strand is 5' to 3', meaning that the 5' carbon faces up in each sugar and the 3' carbon is below. The direction is based on a numbering system assigned to carbons in each deoxyribose in the coding strand. All these sugars are facing upwards. In the non-coding strand, all the sugars are facing down. For the mRNA strand, this is the ribose sugar, and this is the phosphate group. For the coding and non-coding strand, this are the deoxyribose sugars, and these are the phosphate group. And these are the nucleotides.
Now let's talk about the first 24 bases of the enzyme protein catalase coding sequence. So how are the DNA instructions transcribed into messenger RNA? Using the non-coding strand of catalase as a guide, RNA polymerase uses the base pair rule to assemble free nucleotides in the nucleus into a complementary strand of RNA. An example of this is when RNA polymerase reads the DNA-based thymine on the non-coding strand and binds it to a free nucleotide containing adenine. This process continues with cytosine binding to guanine and guanine binding with cytosine. But RNA will never contain thymine, so whenever RNA polymerase sees adenine on the DNA template strand, it pairs adenine with uracil. By using the non-coding strand of DNA as a guide, the genetic code from the coding strand of DNA has actually been transcribed into messenger RNA. When transcription is complete, the messenger RNA, which is small enough to fit through a nuclear pore, takes the genetic code out of the nucleus to the ribosome, the site of protein synthesis. We made this model to represent a small section of the gene for catalase. These letters, the order of the gene, give the instructions to make the protein catalase. We found its CDS origin and took the first 24 letters to create our model.